Hi, my name is Rebecca Moore, and today I'll be talking about the utility of recurrent neural networks in the field of neuroscience. I will go over what a recurrent neural network is, how they are used in neuroscience research, and what they can tell us about the brain. So what is a recurrent neural network? Recurrent neural networks are a type of artificial neural network, or ANN, that share some key similarities with the biological neural network of the brain. They receive inputs from a series of units. All the neurons are connected together, and that connection is described by a weight matrix. This graphic shows the input layer that feeds into the recurrently connected neurons, where each neuron has a weight that comes together to form the weight matrix. This is what is changed to adjust the output layer to match the expected output. Some key features of RNNs. Typically, they are nonlinear. They are recurrently connected. They have a weight matrix, which describes the strength of connection between the two neurons, like synapses of varying strengths in the brain. They can do tasks by taking an input and passing it through the recurrently connected layer for a period of time, and this layer then projects onto the output units. The output is compared to the desired output, and then you compute the error and adjust the weights to iteratively reduce the error. That is a trained network. They can do complicated tasks, especially those that involve time, such as tasks that require memory to be stored. Here we see the equation for an RNN that describes how the activity or the firing rate of units and the recurrent network change over time. If we break down the parts of this equation, we have the left side, which is the derivative of x over time, and then we have x, the vector of the activity of the recurrent units. And at each time step, the activity of each unit is added to a weighted combination of inputs from all other units, or j phi x. The inputs are passed through a nonlinearity. We need this nonlinearity to get rich dynamics or changes in activity over time. There can also be periodic inputs h added to get most favorable dynamics. So why would we want to use RNNs in research? Parts of the prefrontal cortex are highly recurrent within itself, and we don't have a great understanding of this area because responses are heterogeneous. To help with this, we use an RNN that is trained to perform the task at hand and then dissect the artificial network, because it's a lot easier to dissect an artificial network than it is a biological one. If we start with a targeted question about what a population of neurons needs to do in order to perform a task, train a neural network to perform that task, then look at the network to see how it did it, to see if it's informative about how the biological network would do the same task. So in the next couple of slides, we're going to be looking at one application of an RNN being used to test two hypotheses to determine which is the more likely scenario. So in this example, the monkey was trained to perform a ready, set, go task while extracellular recordings were made from the dorsomedial prefrontal cortex, which is an area of PFC. The monkey was required to measure a period of time, TS. Depending on the context indicated by the color of the fixation point, the monkey needed to saccade to a target at a time stipulated by TS multiplied by context gain. Context gain was either 1 or 1.5, and then here we have the target interval TT. So there are a few critical things to keep in mind. The TS varied, meaning the monkey needed to measure intervals that were of different length and the context would change. Over here, you can see the monkey did this task well. TT, the expected interval time, is this dotted line here, and then we have the circles, which are the actual behavior that the monkey did, and we can see that they line up really well, meaning the monkey did the task well. Now the question is how the individual neurons or the population of neurons in DMFC give rise to this observed behavior. To do this, let's take a look at individual unit tuning and how in a population summary of activity and how they change with stimulus and context. So this figure shows the neural responses from the DMFC during the task. These are the firing rates of 5 example units during various phases of the task, split by context G and the measurement period TS. The left is the ready, the middle is the set, and then we have the go. Here you can see the heterogeneity in different dynamics. The gray lines are activity during the gain factor of 1, and the red are during the gain factor of 1.5. So some takeaways from this are that the activity of single units sometimes differ between context. See how sometimes the gray lines are different from the red lines? But overall, you can appreciate that these responses differ from unit to unit and by time, showing how the responses are heterogeneous. The question is then, how does the population of neurons support the timing task behavior observed? Here we can see that the population activity during the ready set phase. The gain axis shown to the left is the axis where the responses were maximally separated with respect to the context gain. We can summarize population activity as it evolved in time by taking the firing rate of each unit at a period of time as being a single point in space. Doing this for each point, we get what you see there. Each point represents a summary of population activity at a given time, and what you can appreciate is that the gray and red trajectories are separate in space. This means that before the monkey needs to generate a time interval TT, the population activity differs between the two contexts. And then by doing the same population activity summary as in B, but now during a different period, you see that the activity follows very similar trajectory, but is separated by context. This is also a clue that there is something interesting about the dynamics of the population activity. So if the population activity starts and ends in similar places in space, activity must be evolving along these trajectories with different speeds. So from this, they created two different RNNs, one where gain information was provided by the magnitude of a tonic input, and one where gain information was provided by the magnitude of a transient pulse before the ready cue. The tonic input RNN was used as a direct test of whether a network with a context-dependent tonic input could emulate the geometrical structure of the responses in the DMFC. The transient input RNN tested whether the structure would naturally emerge in a network without such an input. So now we're going to take a look at the hypotheses that they proposed. So for the gain-independent hypothesis, or the transient input case, G and TS, the sample interval, are specified by the initial condition X naught. Thus, you get a spread along the initial condition axis. They're all spread across here. After the set condition, these open circles, activity evolves towards an action triggering state that crosses over here with a speed, the colored arrows showing through. In contrast, for the gain-dependent hypothesis, or the tonic input case, this is where the input specifies the context, meaning population activity is initialized in the same place. The initial condition encodes TS, or the sample interval, not TT, the target interval, which is why some gains appear to be overlapping. As you can see here, they're not spread down the initial condition, they seem to go together. Then finally, this shows the DMFC data. The top is the unknown mechanism here. The bottom shows the projection of DMFC activity. And then the very bottom shows that neural trajectories form two separate sets without altering their organization. So next, we're going to take a look at the schematic of the RNNs and the outputs for each hypothesis. This is an illustration of the RNN trained to do the same task as the monkey. The network receives a context-dependent input that either stops before the ready signal, which is the transient input, or continues through the trial, the tonic input. The networks were trained so that the output, Z, would generate a ramp as shown in the red lines after the set Q that would reach a threshold, the dotted line, as the context-dependent time. These top two graphs show the state space projections of the tonic and transient RNN activity in the set go context. Here we see the tonic, and then here we see the transient. And then the bottom graphs are the top ones just rotated to make it more clear what their space is. So then some conclusions that we can draw from all of this data that we've seen. The results indicate that the flexible control of behavior can be achieved in terms of systematic adjustments to initial conditions and sample external inputs of a dynamical system, meaning neurons receive the gain information as a context-dependent tonic input. So we can see over here. This was proved by the RNN models because only the tonic input RNN could reliably establish and maintain separate structures of neural trajectories across gains, similar to what we saw in the dorsomedial frontal cortex. Here we can see that they look very similar. Overall, RNNs have more found their place in neuroscience as a post facto tool used as support, not as primary evidence. With the fast developments in computational neuroscience, it could be possible that a more advanced RNN is created that more closely resembles the brain and takes into account the biological constraints, such as how much space you have, how many neurons we can fit, the information available to neurons. At that point, RNNs could be a much more valuable tool used to test proposed mechanisms without needing a fully formed hypothesis first, but as they stand now, that is not the case. Thank you for listening to my presentation.